Right. So when Peter Moody asked me to be part of CCP4 Working Group 2, I didn't imagine that then being in that group of people, I would then be tasked to open this section of the weekend, but I'm delighted to do so. And I was tasked to give a sort of introductory talk on refinement. And I am in two minds about this, because on one hand, I understand that I want to speak to the junior structural biologists in the audience who may not actually be dealing with refinement, so that they have a sense of context and then link out to the general theme of the meeting, and I will try to do so. And the outline of the talk indeed starts with, or rather, it's mostly about refinement in the context of single crystal X-ray diffraction data. I'm hoping that the colleagues who are and friends who are more experienced will not mind, and I'm hoping that if anything else, this serves as a sort of list of references for the junior people in the audience. I'll go back to this one because I have actually asked Dale Tronrud to help me with this, and he's been wonderful. I have not really been developing software for more than 15 years, so you can imagine that I'm skating a little bit on thin ice. At the end, I will just address the fact that refinement these days is thought of in a bit of a broader context. And I have gone as far as risking making a list of the 10 things I rate about refinement. So that's my last slide. So when we think of refinement in the context of single crystal X-ray diffraction, we think of it as a stage in an iterative process, which comprises refinement per se and model building. Obviously, the crystallographer, in this case, macromolecular crystallographer, starts with an electron density map, which requires phased structure factor amplitudes, and we will not go on where these phases come from. And as soon as you have a map, you can build a model. Perhaps you build it manually, perhaps you use an auto builder. And at this point, you have refinement, i.e. an process by which parameters in the structural model are varied and optimized to match a target function of the observations. And in here we have a arbitrary surface. You can see that there are several maxima, several minima, and upon going from one local maximum or minimum, depending which way you look at it, Dell wants to say minimization, okay? Minima, you want the overall minimum, but you are trapped in a local minimum. Can you go there without climbing a maximum? That's not always the case. So we have the obvious radius of convergence problems that all optimization problems may have. Once you've moved the parameters, you shifted the parameters in your model, you can compute a new set of phases and a new map, and you go back. And here, perhaps I will not touch on this any further later on, but I want to bring up some sort of dream that we had, several of us, I think, years ago, that at some point refinement and model building would be integrated in our graphics program, and we would be able to refine the structure on the fly as we build it, and we would see the updated maps, so that we would always be building in maps that come from phases from refined models. That hasn't quite happened yet. So we mentioned that refinement in macromolecular crystallography uses observations, and in fact, I put it in quotes because sometimes we use information that's not per se being measured, but broadly speaking, we will have done a diffraction experiment on a single crystal, so we have indexed it and we get our unit cells. We want to go into symmetry, and we measure our structure factor amplitudes. Perhaps, though, we are going to use during the refinement some so-called experimental phase information, external phases, and those will come from having measured isomorphous or anomalous sets of differences and having interpreted them with whatever causes these differences. It could be, for example, a heavy atom or a set of heavy atoms. Broadly speaking, there's more information that goes into refinement, and we can think, for example, of the fact that most of the time we know the sequence of what we have in the crystal, be a piece of DNA, RNA, or protein. And we have a lot of knowledge about ideal geometry, on which we will touch briefly later. And we know very well, for example, not only amino acids, but we know secondary structure of proteins. We know that the crystals contain the mother liquor in which they grew, and in a more or less disordered fashion. Each of these observations would optimally be dealt with by knowing or by modeling its probability distribution. So if they are Gaussians, we can specify it by an estimated standard deviation for the Gaussian. But in the case of external phases, it will be encoded in a set of Hendrickson-Lattmann coefficients, for example. Obviously, not all the prior knowledge that we use is continuous and therefore amenable to have a probability distribution. What are the models in our, what are the parameters in our models? Well, at least, or very simply, it's a structural model, it's structural biology, so it will be atoms, and we have a certain number of them, and we start modeling an occupancy, which essentially models the fact that sometimes some place in space is occupied by a certain atom in some cells of the crystal, but not in all of them. Typically, when you have alternate conformations of amino acid side chains, or ions bound, or ligands, and not in full occupancy. The positions, of course, and then there are parameters that are meant to model thermal motion, harmonic thermal motion around equilibrium positions, but end up perhaps mopping up some static disorder, and they can be isotropic or anisotropic. And just to remind yourself, I have here written down how they specify the thermally smeared electron density, which comes from the convolution of the static density and a temperature factor, and therefore here are the static density and the temperature factor. Here I will touch upon one aspect, which is that very often we refine models that are not complete, and therefore we have to deal with refinement in presence of incompleteness. And I worked on this some time ago, and that sort of attempt at modeling refinement in presence of incomplete models has not been followed up. When it comes to parameters, there's not only atomic parameters, but there's also overall crystal parameters. For example, scaling parameters that relate the observation which are not on absolute scale with the calculated values for the same observations which are on absolute scale. But you have modeling of the solvent and so on. And more importantly, these days, given we are dealing with likelihood methods by and large, there are model imperfection parameters which sort of allow us to judge how good the model is at the stage of refinement we are at. Target functions, we can use various programs. This table is by no means exhaustive, and by preparing this talk, I realized that I was going to offend somebody by omitting them or by misquoting their work, and I'm prepared to be attacked in my private room after the talk. But you have four sort of, broadly speaking, types of target functions. The field started with the first two, and then maximum likelihood came of age in the late 1990s. Real space refinement is another way you can refine models by matching the electron density in real space. Optimization methods, obviously, as we already saw the picture, and we need the values of the target function for the current model during refinement, and we can compute the gradient, and we can compute the matrix of the second derivatives of the target function. Not all algorithms use both gradient and Hessian, and in fact, full Hessians are rather rare in macromolecular refinement. We have, as all fitting in science, the problem that we may not have enough data to determine the parameters. And here I have a standard example which shows that with such a protein and 5 parameters per atom, we are already in trouble. And Axel Brunger was the first one who tried to address the problem of how to decide whether we are over-parameterizing our refinement, and introduced R work and R3. You can read about it in this paper. And I tend not to allow my refinements R and R3, R work and R3 to go further apart than 5%, but this is actually an arbitrary and it's not cast in stone. So you can fix certain parameters or tie them to each other. And in this way, you start losing degrees of freedom, which is where the overfitting risk comes from. And in here, I've listed a few types of refinement that do use constraints. But more interestingly, you can add extra observations in the forms of restraints. And the most important ones are ideal geometries that come from dictionaries, and they add a term to the part of the target function that fits the observations. And then you are immediately asking, where does the value for this weight come from? And methods sort of optimize the relative weight of target function that fits the data and the one that fits the ideal geometry. 2 slides on ligands. These days, when we have a ligand in a protein, of course, we need a stereochemical dictionary for it too. And there's a glorious history of automation of creation of such dictionaries in a CIF format these days. Be careful that you look critically at your dictionary. And in here you see FAD, which in CPK model is obviously planar. And you can also see that if you go to FAD H2, adding hydrogens on these nitrogens, you to add a hydrogen here, keeping the molecule planar because of clashes. Unfortunately, some automatically generated dictionaries, like the one in the picture, we will not blame the program, do create planar FADH2 dictionaries. So be critical and use your chemistry. Other types of restraints have been introduced throughout the years in order to manage the problem without avoiding overfitting. And I will not go into details of this. But the trouble is, every time we introduce new restraints, we end up causing some correlations. So the fit to the data, every piece of data drives refinement of all atomic coordinates, for example. But if you have a restraint between 2 atoms, that will introduce correlations between moving them. And if you ignore the Hessian terms that tell you that you could move both of them in a certain way to maximize your target, to optimize your target function, you are in trouble, because they are effectively moving independently. They're supposed to move independently, but they're not independent anymore because there's a restraint between them. So the complexity of the refinement landscape increases. And here I'm quoting this recent beautiful paper by Kevin Cowton. And using high order derivatives of the target function can only partially solve the problem. The other interesting aspect is that when you don't use a full Hessian, you end up having problems in the reaching convergence. And so much so that you would actually get where full Hessian matrix refinement will get you in so many steps that then numerical aspects of the procedure come into play. So recently, or not so recently in the case of this one, alternative approaches have been tried to avoid or to address these problems. And in particular here, I want to point you to this work by John Aguirre and Kevin Cowton, who using difference maps, gradient and Hessian, of the electron density with respect to the parameters, sort of propose a different approach that can actually work also to change maps directly rather than atoms. Another trouble with geometry restraints is that in order to compensate for the incorrect assumption that the restraints are independent, then we introduce extra restraints, for example, planarity or chiral volumes. And so these restraints that we use end up being redundant. And in this picture, Dale shows on a Ramachandran plot, the values of two of the angles around the carbonyl and peptide bond, and you can convince yourselves that the C alpha CO angle and the C alpha CN angles are anticorrelated. So in red you have values that are smaller than 120, and in blue you have closer to 120 in red and much smaller than 120 in blue. One aspect of refinement that has not been paid enough attention, I think, is refinement against several data sets. This is typically the one example that I found in the literature, which is a piece of work by Dale, who refined simultaneously against several data sets of a certain protein with many inhibitors. But of course, there's also the question of refining against data sets of different polymorphs of the same protein. Much in the same way we do multicrystal averaging against polymorphs. Here, I will not have to convince you with too much difficulty that in the PDB, we have structures that use multiple methods these days, and you can see that this is on the up. So we come to the question of what's the role of refinement and what's the shape that refinement will take in this context of integrative structural biology, and where we have observations, for example, like cryo-EM maps, electron diffraction data from techniques that help us finding out which subunit of a complex are close to each other, and so on. Well, such models are slightly more complex than models for molecules in crystals, and we are left with the choice of what to optimize. And obviously, each of these different kinds of data will have to drive the refinement of these models with a different weight, and it's not trivial to assess how to weigh the various terms, plus some of them are kind of difficult to parameterize or to throw into the function. Very recently, I had a very simple task of fitting my crystal structure, or rather, yes, I fitted a homology model built on a crystal structure of mine in a 25 angstrom negative stain EM map of a homologous protein plus a fragment antibody. And I was looking for a piece of software that would do it. I ended up doing it in Chimera using fit to the local density, and then using the information that I had on the epitope to validate. But obviously, you could have decided that this was a piece of data that was driving your fit. So in conclusion, 10 things I rate about refinement. One is current numerical implementations probably do not make the best use of currently available CPU power and memory. And this idea of on-the-fly real space, reciprocal space refinement during model building ought to be possible. Improvements to convergence will need to address better treatment of correlation in model parameters. And perhaps the solution there is either proper treatment of Hessians or joint likelihood functions that obviously score the likelihood of a model against more than one structure factor amplitude. So introducing correlations between them. And this will address problems that are caused by geometry restraints, redundant geometry restraints, non-crystallographic symmetry, multiple polymorphs, and so on. I've already mentioned incompleteness, and then the worlds are oyster now with writing refinement programs against multiple data sources. And here I want to thank Del for all the support. He's probably there in the middle of the night or the early hours of the morning in America, and the organizers, the people who taught me refinement, and George Sheldrick, who always helped me with Shellix. Thank you.